Good evening, ladies and gentlemen, and welcome to this session organized by Resono Ophthalmic, the Italian company behind Rexon-Eye. My name is Alfredo Ruggeri. I am a co-founder and chairman of the company. In this event, I will first talk about who we are, which is our background, what is our instrument Rexon-Eye, and the technology on which it is based. I will also give you some information about the clinical evidence we have gathered so far about the device and what is ahead of us, so what we expect to do in the next months or years. In the second part of this event, Dr. Luca Vigo, head of the dry eye section at the Carones Vision in Milan, will report about his own experience with the device and the results he obtained. He is one of the earliest adopters of Rexon-Eye, so we have pretty much to say about this. As regards my own presentation, we will first say something about what is our background, briefly about dry eye, the QMR technology, which is at the core of our instrumentation, how this QMR technology was applied to develop Rexon-Eye, the clinical evidence, and what we believe is in the future, is reserved for the future for ourselves. So who we are. Resono Ophthalmic was founded very recently in 2014, but it is actually the last step of a long personal journey in science, technology, and entrepreneurship. Up to last March, I was a professor of biomedical engineering at the University of Padua, and with my group there, we developed techniques and software to analyze retinal and corneal images. However, very soon, I realized that for me, it was very important to apply the results of our scientific research to the real world to help and assist people. So I started to cooperate with companies, and the first one was a 10-year-long cooperation with NIDEC and the NIDEC Technologies, which is based in Padua, to help them to develop their instruments. Then a twist to this move towards exploitation of research in the real world came when, with some colleagues from university and industry, we established M31, a private incubator under which several successful companies were created: Centerview, Adaptica, DI, just to mention the ones in ophthalmology. At the same time, I was also cooperating with another company, Telea Electronic Engineering, and they were working on a breakthrough technology that would later become the QMR technology. So it was meaningful for me to merge all this and to create a company to apply QMR in ophthalmology. And then Resono Ophthalmic was born. Of course, I will not dare to explain to you what dry eye is. I will only quote the definition by Tifos Due2 report and mention that you may go from mild discomfort up to severe corneal disorders. And what is important is that this disease is very common. There are hundreds of millions of people worldwide suffering from this, and it is also increasing with time. There was a recent paper by Reza Dana saying that overall prevalence and incidence are increasing over time for all demographics. And this is not a trivial cost for the society, because back in 2011, the overall burden for the patient was $5 billion only in the USA, and the cost for the whole society was almost 10 times that one. several options are available to treat dry eye, but unfortunately most of them, or only all of them, only alleviate symptoms at short or mid-term and do not address the real cause of the disease. So recently Rexon-Eye came up. I would like to call it the new kid in town, if you're familiar with the Eagles song. So it was a newcomer in the area of dry eye. It works by generating and delivering to the patient an electrical signal applied by using a mask. And this electrical signal, we will see in a minute, is able to stimulate the natural regeneration of cells and tissue. And for this reason, this therapy is effective in both evaporative as well as aqueous deficient dry eye. It is a non-invasive treatment, it's actually a pleasant one, with long-lasting symptom remission. I would like to explain to you why electricity. We know very well that electricity is the way by which we are able to see, because the stimuli goes into the retina, are transformed into action potential, and they go to brain. At the same way, our heart is able to beat because an electrical signal stimulates the muscles, either an endogenous signal, or in case of cardiac failure, a pacemaker goes to stimulate the muscle of the heart to beat and to allow the patient to survive. Or in functional electrical stimulation, we are able to have a paraplegic patient to walk by stimulating in a proper way and proper timing all the muscles of the leg. And very recently, just this August, a paper came out on IOVS about the effect of electric field to direct the growth of retinal ganglion cell axons. So it is by now quite clear that electrical signals are a key ingredient in most, if not all, the functions of our body. But the question is, what unique electrical signal can we apply to stimulate the natural regeneration of tissue, and so, in our case, to treat dry eye? Well, to explain this, we have to go back in time to the 80s, when the problem was, how can we perform an electrosurgery without increasing the tissue temperature? You know that electrosurgical unit works by applying a current through the handpiece. This current here increases the temperature of the cells. The cells explode because the intracellular fluid boils, and you are able to obtain a cut effect. But of course, this was exactly due to the fact that you deliver heat. So it seems that obtaining a cut without increasing the temperature is an almost impossible task. But actually, there is a solution. And the solution, the very bright solution, was resonance. What is resonance? Resonance is a very well-known physical condition. You see here an old movie of the Tacoma Bridge just south of Seattle. One day the wind was blowing, not particularly strong, but it was blowing exactly at the same frequency of the natural oscillation of the bridge. So the bridge started to oscillate, and eventually it broke down. And it's exactly the same way by which, if you emit a specific sound, you are able to have a glass to oscillate until also it breaks. If you want to break the glass, you have to apply a very strong force. But if you emit a sound exactly at the frequency of oscillation of the glass, a very small pressure is able to make it oscillate. This was resonance. So, back to electrosurgery now. Gian Antonio Pozzato, the inventor of this technique, developed an electrical stimulation that he called quantum molecular resonance, or QMR, which is an electrical current at specific frequencies, in this case 4 megahertz and harmonics. And these frequencies are exactly in resonance, now we know the concept, with tissue molecular bindings, so they are able to open the bindings and obtain the cut without wasting energy into heat. Of course, this allows you to obtain a non-traumatic cut and a soft coagulation, always at temperatures below 50 degrees. And this have a lot of advantages from a clinical point of view. No thermal damages. You can apply this for biopsy, neurosurgery, absence of any choroid tissue, and for the patient, reduction of postoperative pain and acceleration of recovery. Here you have a picture of a cut. You see this is a perfect cut. Nothing is charred, nothing burned. And here I have a stunning movie showing a surgical intervention with this device on the dura mater membrane inside the brain. That would be totally impossible with any other electrosurgical unit because you would boil the brain. This is possible with this specific device because you can cut without increasing the temperature. So, after these results, Gian Antonio Pozzato founded his own company, Telea Medical, and he patented, of course, all this, and then they developed a line of electrosurgical units for the different disciplines of surgery, and these are sold all over the world. Then serendipity played a role, and so they discovered that the same kind of stimulation at a much lower power and with a slightly higher range frequency is able to be effective in stimulating the natural regeneration of cells and tissue. And several phenomena can be used to explain this effect. This electrical field is able to change the cell shape. There is an increment in the calcium release, and we know that calcium is a messenger in many metabolic processes, and there is also an increase in the adult stem cell generation. And these results are described in a paper, and that picture shows a section of cutaneous muscles in rat where the presence of parvicellular infiltrates, they are a clear sign of the regenerative process. Recently, there was another study published on this PLOS ONE paper. Human mesenchymal stromal cells were treated with QMR, and a complete microarray analysis was carried out using the techniques of cDNA, and transcriptional changes were found on the treated cells. In particular, there was an enrichment of several processes and pathways, including metabolism and cellular organization, and some genes were upregulated, exactly those genes involved in the extracellular matrix remodeling and wound healing. So, after this, Telea Medical decided to have a line of tissue regeneration devices for medical application, and they are now applied in physiotherapy, aesthetic medicine, pain medicine, and wound healing. And there is another paper of a trial at the hospital in Turin where they treated 11 patients with very, very severe limb wounds, and the device was able to treat them and to have an improvement in the granulation of tissue and wound contraction, a significant reduction of pain for the patient, a decreased amount of inflammatory cells, and in general, a significant improvement and acceleration of the healing process. In this picture, you can see one of these wounds. They were not able to treat this wound with any other technique, and then after 10 treatments, you can see that the wound is closed, but it's closed not with fibrotic tissue, it's closed with functional tissue, so you can see the re-epithelialization island in the center of the wound. So this is very interesting, very important. So the idea was to bring this to ophthalmology, and so to exploit the QMR regenerative effect in ophthalmology, and in particular we chose dry eye as the target pathology after some empirical evidence of this effect. And this is the first device, Rexon-Eye Model 1, that we applied in the first trials. You can see that the electrodes are placed around the eye so as to stimulate the ocular region. And we did the first exploratory trial at the University Hospital in Berlin on 7 subjects. 6 out of 7 exhibited improvement in one or more of the typical indexes used for assessing dry eye. And 1 subject in particular moved from 25 eye drops per day to 0, and was 0 still 3 years after the end of the therapy. This table summarizes the results, and this line refers to this very lucky lady who could stop his dry eye therapy with drops. Then we decided to move with a formal trial carried out at the University of Verona on 27 subjects, same protocol. Here we were able to group the patient in evaporative and aqueous deficient patients. Follow-up was 12 months after the end of the therapy, even if we have data up to 18 months after the end of the therapy. And this is a slide with the table for results for OSDI. You can see that even after 1.5 years, you still have an improvement in the OSDI parameter. The same for corneal staining. But I think that very important is this slide, where you can see that for the 3-year backup time, the average value of this parameter was still improving 1 year after the end of the therapy. And the same happens for the Schirmer test. So in our view, this is an evidence that you are treating the cause of the device and not just alleviating the symptoms. Out of this trial, we published a paper on the British Journal of Ophthalmology, where you can find all the details about the trial protocol and the results. So after this very encouraging result, we established the company in 2014, and we designed a new device for the specific application. Now it's a tabletop device, very easy to use it and set up. Then we thought, since we have to deliver this current to the meibomian glands and the lacrimal glands, which are the glands involved in the tear production, we need to exactly have current flowing through those glands. So we need to design an electrode which can be placed not around the eye, but over the eye. And so we designed this new electrode, which is kind of goggle. And you see here, number 3, is the actual electrode, which is powered by the circuit, and we have an outer and an inner shell. So this is the inner part and the outer part of this goggle. Then we designed the graphical user interface in order to allow the operator to easily use the machine. And we were able to move from the old protocol with Model 1, let's say, which required 12 sessions over 2 months, a bit heavy for the patient. We were able, with Model 2 Rexon-Eye, to have a new simplified protocol, 4 sessions over 1 month, so 1 session per week, and the patient sits there enjoying the treatment for 20 minutes wearing the mask. Rexon-Eye is a CE-marked device as a Class 2 device, as a medical device for the treatment of ocular surface disorders. We also did some intellectual property protection, so the device is protected by a patent issued in Europe, and now also by a United States patent issued just last month. And of course, we have several other patent applications pending in many countries around the world. In order to provide clinical evidence of the efficacy of this device, we did at the San Raffaele Scientific Institute in Milan an experiment on animal. We took 40 mice, and they were all subject to alkali burns. We treated half of them to see how this treatment is able to protect against ocular inflammation. And the results were that you have a 50% reduction in corneal perforation in the treated mice with respect to untreated mice. And I presented these results at the ARVO conference last year. In the same institution, San Raffaele, we also did a clinical trial. The objective was to study the efficacy of this device on dry eye patients and specifically meibomian gland disease patients. The treatment protocol is based on model 2 Rexon-Eye, so the simple one, 4 treatments, 1 per week. And in addition to the typical parameter, we also measured the meibomian gland expression and the meibum quality in order to be able to assess the improvement in the functioning of the meibomian glands. And the key point, we measure at baseline and 1 month after the treatment. And here you have the summary of the results. And I think it's very important to stress that all efficacy endpoints had a statistically significant improvement. Both the meibomian quality with a very strong statistically significant, but also the other ones. So all of them improved on average and for most of the patients. And we report about this clinical trial on the paper just appeared on Cornea. Of course, we have in our way convincingly shown the efficacy of Rexon-Eye in published clinical trials. However, we need to strengthen, widen, and diversify this clinical evidence in different populations around the world, but also in different pathologies. So the idea is to start several programs which are already in progress or about to start worldwide. We have one trial going on in Germany at the Potsdam Eye Hospital. It is based on 70 patients with keratoconjunctivitis sicca, and we hope to have the results still ongoing. So we hope to have the results by the end of the next year or mid of 2021. But we have also several other trials or patient testing around the world, some also to help in local product registration. In Europe, Italy, Greece, Spain, Germany, and Poland. In Asia, we have one trial just started in Singapore, in Hong Kong, and South Korea, and also in Latin America, in Brazil, and in Chile. So I will say that the new kid in town has been performing quite well so far. The key point, in my view, is the scientific evidence of the stimulation of the natural regeneration of tissue, which is able to address the cause of the disease and not just the symptoms. For this reason, it has been shown to be effective in both evaporative as well as aqueous deficient dry eye. It delivers long-lasting symptom remission, and it also, since it is a quite general treatment, it has promising application also for other ocular diseases. We will soon start a trial also to assess the efficacy of this device in other ocular diseases other than dry eye. So in summary, I will say that very exciting times are ahead of us, so please stay in touch with us at Resono Ophthalmic through our website or our LinkedIn or Facebook page. Many thanks for your kind attention.